Everyone wave and say hi.
Welcome to the Heinrich-Corless Research Labs, where we study gastrointestinal stromal tumor and improving therapies for patients. In the Heinrich Lab, we perform drug testing to test the efficacy of new drugs in GIST cell models. GIST cells that have been isolated from patient tumors can be grown in plastic plates or flasks inside an incubator and observed using a light microscope. These GIST cells have biology similar to the cells in tumors and can be used for testing drugs. The cells can be transferred into plates for different experiments inside a sterile hood. The mutant proteins from these cells, like KIT and PDGFR-alpha, can be collected to test how well a drug works. We visualize mutant proteins using a technique called Western blotting to determine the best dose for each mutation. This is how treatments like Gleevec, Sutent, and Stravarga were originally tested against GIST mutations before being approved. In the Corless lab, we can identify gene mutations from patient tumors that have been collected during surgery and fixed on slides. Tissue is scraped from tumor slides for isolating DNA for sequencing. The DNA from patient samples is isolated, purified, cleaned, and enriched in preparation for loading onto the sequencer. DNA samples are dispersed on a microchip and loaded onto the personal genome machine for sequencing. Within a few hours, the data is available for analysis. We can determine, for example, if a patient's tumor bears a KIT exon 11 deletion or point mutation or is wild type. Molecular diagnostic information like this is used to design treatments for each patient. There are also projects that aim to discover new things about GIST biology. We can incorporate data from previous research and clinical trials to ask and answer new questions about the nature of GIST growth and drug resistance. Graduate students in the lab work on individual projects in GIST models with potential future clinical applications. To discover new drug combinations, we utilize high-throughput robotic assemblies to plate large numbers of experiments at once. We have also used these technologies to identify novel drug targets in KIT mutant GIST cells. Using simplified models, we are investigating the consequences of different GIST mutations, such as SDHA. By understanding how each mutation affects the biology, we can get closer to developing specific treatments for patients with these mutations. Doctors Chris-Corless and Michael Heinrich lead our dedicated group of scientists working on GIST research. GIST research is important to us, and we are motivated by the potential of what we discover to improve the lives of GIST patients. GIST Cancer Research Fund support has been invaluable to the Heinrich and Corless labs and has made our research possible. Sincere thank you from all of us at the Corless Heinrich Research Labs for all your support.
Thank you, GCRF.
Thank you. Thank you for the support. We appreciate it.
Thank you.
Thanks, GCRF.
Thank you.
Thank you.
Thank you for the support.
We really appreciate the support of the GCRF. You guys are making all the difference.
Thank you.
Thank you, GCRF.
Thank you for everything.
Thank you for your support.
Thank you, GCRF, for supporting our work and for truly being our partners in this endeavor so that we can treat and cure this disease.